Afternoon, everybody. My name is Alan Bernstein. I'm president of the Canadian Institutes for Advanced Research. Before I introduce the first speaker this afternoon, I would like to first recognize John Dirks for his tireless dedication to the Canada Gairdner Awards program and his vision of the Gairdners, as they are now called around the world, as not just an opportunity to give out prestigious prizes to truly outstanding health researchers, but a national event involving Canada's entire health research community, young people from across the country, and a vehicle for showcasing to the world and to our politicians the importance of research for improving health and our well-being. I remember well the meeting that John and I had when I was president of the Canadian Institute of Health Research in my office, in which we discussed this sort of broad, exciting, compelling, and ambitious vision for the Gairdner program. And to John's great credit, he successfully pulled it off. And John, so all of us in Canada's research community owe you a huge debt of gratitude for all your efforts on our behalf. Thank you. So now I'd like to turn to this afternoon's Gairdner Awardees. This afternoon we have sort of one of each. We've got an outstanding Gairdner International Laureate. We have a Gairdner Whiteman Laureate and the Gairdner Global Health Laureate. But despite this seeming sort of potpourri of awardees, I think they have a lot in common. In fact, I think they have at least 4 things in common. First, they all share a passion, a true passion for science and for the importance of science, both to understand our world, but also to improve human health. Second, all 3, I think, share a passion for people, colleagues, trainees, and their mentees. Third, all 3 struck out on different paths not at all previously well-trodden in their own chosen fields of study and activity. And fourth, I happen to know all 3 Gairdner awardees this afternoon, but each in very different ways. And I'll come to that when I introduce each of them in turn. Now, I'm not going to read their extensive bios. That's in the booklet that we've been given, or the work that led to their awards, because I think we're about to hear from them about their work, so you don't need to hear it from me. But what I'd like to do is give you some personal insights into each of the awardees this afternoon. And I'll start with Lynne Maquat, our first lecturer. Our first awardee is from the University of Rochester. She's been working for over 30 years on global mRNA expression and RNA expression more broadly, and protein expression as a result, and more specifically on nonsense-mediated mRNA decay, or NMD, a phenomenon that she first reported during studies of globin expression in people with hemolytic anemias and beta thalassemias. To me, Lynne's research illustrates, I think, the power of science. I don't think anyone would have predicted that NMD would have existed. There was no need to think about the nonsense mutations in the genome would affect post-translational or translational issues as opposed to just translation itself. And yet it does, and you'll hear, I'm sure, something about it from Lynne this afternoon. I first met Lynne in the 1980s at a Gordon conference on red cells. We met under very unusual circumstances which actually I promised Lynne not to reveal in my introduction. Or ever, okay. So but I can say that it was in a dormitory. You know, the Gordon conferences. Gordon conferences are in private schools in Upper New England, and Lynne made the mistake of assuming that there was a girls' dormitory and a boys' dormitory. So it was not a safe assumption. So that's all I'm going to say, because I made a promise to her. Lynne has been a very strong champion of women in science. And I just want to note in passing that 2 of our 3 awardees this afternoon are women, and I think it's about time. Lynne, over to you.
We'll talk about that later. Thank you, Ellen. It's always a pleasure to see you, and I feel very honored and privileged to be here talking to you today. I don't know if you can see Mahakala. He's a wrathful deity in the Tibetan tradition, and he represents the goodness of destruction. He destroys our 5 kleshes, which are our 5 negative afflictions, and I'm going to talk about a pathway that is destructive and in part also for our own well-being. The pathway is called nonsense-mediated mRNA decay, which we abbreviate NMD. By way of review, I think you know that translation terminates at one of 3 nonsense codons, which are shown here in red. These codons usually don't encode any amino acid, and instead signal the release of the protein that's being made, and ultimately dissociation of the 2 ribosomal subunits from the mRNA template. Many years ago, we realized that a number of human diseases could be due to frame shift or nonsense mutations that result in the premature termination of translation. And the disease is due to the absence of full-length functional protein. But as Alan said, we also realized that if translation terminates sufficiently prematurely, then the mRNA will be targeted for NMD. And we first reported this in a paper in 1981, studying the beta-zero thalassemias, which are a hemolytic anemia, as Alan said. And then my first graduate student, Ira Dar, who now has been for many years senior scientist, a lab head at the NCI, recapitulated studies by studying another hemolytic anemia and a neurologic disorder called triphosphatidylisomerase deficiency. The reason why NMD exists is because truncated proteins that are produced from mRNAs that prematurely terminate translation can be toxic. So it behooves the cell to eliminate their production, and one way to do this is to eliminate the mRNA from which they derive. I think you all know that diseases are heterogeneous at the molecular level, and hence the need for personalized medicine. 2 patients with cystic fibrosis, for example, can have different mutations that debilitate CFTR production. And it turns out that NMD targets disease-associated mRNAs in 1/3 of individuals with an inherited or an acquired disease, such as cancer, and including those causing cystic fibrosis, Duchenne and Becker muscular dystrophies, and many cancers, hundreds of different diseases. This is a very incomplete list of genes that harbor nonsense mutations that cause disease, and note that this is a list that only includes nonsense mutations that cause disease, not the many frameshift mutations that can also cause disease, all of which would result in the premature termination of translation. Note that there are tumor suppressors in this list, and again, it's a partial list. By way of review, this is a very simple overview of how genes that encode proteins in mammalian cells are expressed. In the nucleus, genes are transcribed in a collinear fashion to produce a precursor to the mRNA, a pre-mRNA. And then that pre-mRNA is processed both co- and post-transcriptionally, so the resulting mRNA has a cap at the 5' end, a poly-A tail at the 3' end, and if it contains introns, which are shown here as lines, the introns will have been removed by a process called pre-mRNA splicing. The mRNA is then exported from the nucleus to the cytoplasm, and it's in the cytoplasm that the mRNA is translated. It directs protein synthesis, and it also undergoes degradation. Our studies of NMD have uncovered remarkable links between pre-mRNA splicing in the nucleus and mRNA translation and degradation in the cytoplasm. And those links we call exon junction complexes, or EJCs. These are complexes of proteins that were discovered by Hervé Lier when he was a postdoc in the lab, and we were working at the time with Melissa Moore. They stably contain some of the UPF proteins that we named after their orthologs in Saccharomyces cerevisiae. And we also uncovered a new template for protein synthesis. This was work from Yasuo Ishigaki when he was a postdoc in the lab. We call this new template the protein translation, the pioneer translation initiation complex. And it turns out that NMD targets largely newly synthesized mRNA that has yet to lose the cap-binding protein heterodimer called CBP80 and CBP20 that's acquired by pre-mRNA. If the mRNA had also undergone splicing, had been generated by splicing, that pioneer translation initiation complex will also contain the post-EJCs. And so what I hope you're imagining is that newly synthesized mRNAs are associated with proteins that make it a very all ready to go RNA protein interaction for surveillance for NMD should it need to occur, because all of these are activators of the process of NMD. They typify mRNAs whether or not they are NMD targets. The cell has no way of knowing if the mRNA is an mRNA target until the process of translation occurs. One purpose of NMD, even though we discovered it by studying human diseases, is to downregulate abnormal transcripts that we make all of the time. And for example, the most frequently produced NMD target are spliced mRNAs. And I'd like to give you an example, one scenario by which this can occur. This is a hypothetical pre-mRNA. The alternative exon is in red. Normally it's removed by splicing, so that translation initiates and terminates normally. There's no NMD. However, if that exon made it to the mRNA by splicing, there are two ways by which NMD could occur. One is if the exon had an in-frame termination codon, in-frame with the initiation codon, so there was a premature termination generated within the exon. And the other is if the exon were in the reading frame, but not a multiple of 3 nucleotides, so there would be a shift in the reading frame and the generation of a termination codon downstream of the shift. This scenario is a type of intron retention, and there are other ways by which alternative splicing can generate an NMD target. For example, exon skipping. If an exon were in a coding region, meaning the region that encodes the protein, and didn't consist of a multiple of 3 nucleotides, and was removed, there would be a shift in the reading frame and the generation of a premature termination codon downstream. And also, there's nothing particularly special about the types of termination codons that can trigger NMD. For example, a normal termination codon can be converted to the type that does trigger NMD if there is splicing downstream of that termination codon. And I think you know that in terms of alternative splicing, we have only 20,000 genes that encode proteins, and we need many more than 20,000 proteins developmentally and as multicellular organisms to have our diversity. And so with the flexibility of alternative splicing to generate our protein diversity that we need comes mistakes. And also in terms of 3' end formation, approximately 70% of pre-mRNAs undergo alternative 3' and cleavage 3' cleavage and polyadenylation to vary the length of the 3' untranslated region, which could or could not include an intron that could trigger NMD by translation terminating normally. We know from work that we did with Ben Blenka, who's in the audience, he's at the University of Toronto, that the third of spliced mRNAs in our cells are generally mistakes, and one hallmark of a constructive alternative splicing event is that it is conserved in a developmental or cell type, tissue-specific way, and these are not. And why does NMD exist? As I said, because the resulting protein, it's a truncated protein usually, is often unstable, but if it's not, it can function in dominant negative ways that are deleterious to cells. So it's an mRNA quality control pathway. It's also called an mRNA surveillance pathway. I want to give you an example of what happens when NMD fails to occur, and the example does include the human beta-globin gene. Nonsense codons, which are also called premature termination codons, within the final exon of the human beta-globin gene, result in a dominantly inherited form of what is usually a recessive disease. Here's a picture of the human beta-globin gene. Translation initiates, let's go to the message that derives from the gene, normally in exon 1, and terminates in exon 3. Here is a premature termination codon in exon 3. And this is the type that does not trigger NMD. And I'll explain the rules soon, so you'll see why this premature termination codon doesn't trigger NMD. The mRNA is stable. But instead of producing a full-length beta-globin protein that can form a tetramer with alpha-globin protein and functional hemoglobin that can carry oxygen in the blood, that truncated beta-globin protein can only dimerize with alpha-globin. And this results in ineffective erythropoiesis, ineffective red cell development, and a dominantly inherited form of the disease. What did I just skip? All right, so you can imagine that we were very interested in understanding how cells differentiate termination codons that don't trigger NMD from those that do. And what we simply did was we moved premature termination codons around in a number of mRNAs, and we also moved introns around in a number of transcripts. And we came up with the 50 to 55 nucleotide rule. This rule says that if there's a premature termination codon more than 50 to 55 nucleotides upstream of the last exon-exon junction that's generated by splicing, then there will be NMD. But if it resides downstream, there's no NMD. So you can see why premature termination codons on the last exon of the beta-globin mRNA fail to trigger NMD. We don't think there's anything special about the last exon-exon junction. We think all junctions that have this distance downstream of a termination codon will trigger NMD. And you can imagine how this rule really works, since I've told you about exon junction complexes that we now know. We called them marks for years. Splicing must deposit a mark on newly synthesized mRNA until the first round of translation. And that mark is now the exon junction complex, which is 20 to 24 nucleotides upstream of the junction. And if a translating ribosome were to get out of the purple region and into the blue region, it would physically remove that exon junction complex because of its leading edge, where its leading edge is, and that would make the RNA immune to NMD. So the rule makes sense. It made sense well before we really understood exactly what was going on. This rule would predict accurately if an mRNA had a premature termination codon, but it didn't undergo splicing, no NMD. And we also know that if there is a termination codon relatively early on and then a reinitiation codon downstream, but before the next exon-exon junction, then there will be an inhibition of NMD that depends upon the efficiency of translation reinitiation. All right, now that we know the rule, I want to go back and talk about how NMD protects us from proteins that are produced in a truncated fashion and could be toxic. So even when disease results from NMD-induced protein deficiency, the lack of functional full-length protein, the disease phenotype may be milder than and different from that caused if the truncated protein were expressed. So NMD protects many heterozygous carriers of PTC-containing genes from manifesting disease phenotypes that would result if that truncated protein were produced. This is a really short list. The list goes on and on and on about genes that are associated with diseases when defective and what happens when the mutation is located 5' to the boundary I've talked about, where the premature termination codon triggers NMD. And you already know in the case of beta-globin, there's a recessively inherited form of thalassemia. But if the premature termination codon were 3' to the boundary, a dominantly inherited form of thalassemia. And there are many examples, rhodopsin, for example. If the premature termination codon is 5' to the boundary, you have a recessively inherited form of blindness. But if it were 3' to the boundary, where there would be no NMD, a dominantly inherited form of blindness. NMD is a quality control pathway, but it also has other purposes, and I'd just like to review what they are. So think about it. The cell has evolved a pathway. It's very useful, and it's going to continue to utilize it to the best of its ability. It's under selection constantly. And so the cell also has developed a coupled reaction of alternative splicing and NMD. And this is a pathway that's often used by RNA binding proteins to auto-regulate. I'll give you one example, and this is an example of the polypyrimidine tract binding protein, which is a splicing factor. This is the pre-mRNA part of the pre-mRNA from which PTB derives. Here's the mRNA. Notice that exon 11 is included, and this produces full-length PTB. However, when the cellular concentrations of PTB become too high, there is an ability for PTB to feed back on the splicing of the pre-mRNA that generates it, and by so doing, exon 11 is skipped. That is in the coding region. It's not a multiple of 3 nucleotides. You get a shift in the reading frame and the generation of a premature termination codon in exon 12, which triggers NMD. So this is an example, one of many, whereby an RNA binding protein can auto-regulate by affecting the splicing of the pre-mRNA from which it derives to downregulate its own production. A third purpose of NMD derives from the fact that 10% of our normal physiologic mRNAs are NMD targets. And what is that about? So NMD, it turns out, serves as a cellular adaptive mechanism where cells can regulate the efficiency of NMD, and I'm going to show you examples where the efficiency is downregulated, so the cell can adapt to its changing environment and respond appropriately. What do these 10% of physiologic mRNAs look like? Well, I'll give you a few examples. mRNAs that have a natural upstream open reading frame, a reading frame that initiates and terminates before the reading frame that encodes the functional protein. Myogenin mRNA is an example of that. And myogenin mRNA is a natural NMD target. We've already talked about mRNAs that have one or more introns sufficiently downstream of the normal termination codon. They are natural NMD targets. And we've studied selenoprotein mRNAs, where the UGA codon, if you remember, that was one of the 3 termination codons I talked about in the very beginning of the presentation. In specialized mRNAs that encode selenoproteins, there's a selenocysteine insertion element that will insert the 21st amino acid, selenocysteine, in the spot where the UGA normally would encode a termination codon. And we know the more selenium we feed a rat, the more selenocysteine incorporation at the UGA codon. However, there's always some UGA codon recognition as a termination codon and some NMD. I'd like to provide evidence that the efficiency of NMD can be regulated by cells as a means to adapt to changes in the cellular environment. And the changes, the stimuli that I would like to talk about is work done in my lab on DNA damaging reagents that activate the p53 tumor suppressor pathway. And I think you know that in cases of severe DNA damage, the cells will undergo regulated cell death, or apoptosis. We've heard a little bit about that already this morning. This is work that's been done by a postdoc in the lab, Max Pop. And he has shown that the attenuation of NMD facilitates the response to chemotherapeutics. This is a story about the key NMD factor UPF1. It's an ATP-dependent RNA helicase, and without it, you have no NMD. The idea is that NMD is a molecular valve that enables cells to respond. Normally, with NMD functional, the mRNAs that make it to the cytoplasm and make it onto polysomes to produce proteins are not regulated by NMD. But in the case of the frontline chemotherapeutic that damages DNA, doxorubicin, there's a cleavage of UPF1. So UPF1 isn't activated, and what happens then is not only are unregulated mRNAs on polysomes producing proteins, but direct and indirect NMD targets are also on polysomes producing proteins, and some of these NMD targets are pro-apoptotic. And the response, therefore, is cell death. If we inhibit NMD, we do not cause apoptosis. However, when the cell inhibits NMD, and it's not a complete inhibition, it primes. It's actually a causative contributing to the apoptotic process, so as to prime cells, to push them in the right direction, so that all the other stuff that happens to promote apoptosis is facilitated. I want to show you some data, just a little bit. This is a Western blot of UPF1 protein, our key NMD factor, showing times of exposing human breast cancer cells to the chemotherapeutic doxorubicin. And you can see as a function of time, we start to see cleavage products of UPF1 occurring. these cleavage products are certainly at sub-stoichiometric levels relative to full-length UPF1, and they are underrepresented relative to full-length UPF1 here in terms of abundance because they are missing regions of the amino terminus of UPF1, and our antibody reacts to the amino terminus of UPF1. All right, so doxorubicin treatment, DNA damage, results in UPF1 cleavage, and that cleavage is conserved. We see it in all cell types that we've looked at, the cow, the hamster. Here's an example of mouse. We see it in dog. And the response occurs when multiple insults that damage DNA are exposed, provided, like the etoposide, topoisomerase inhibitor, and also cyclohexamide. So you can see as a function of time, we see the UPF1 cleavage product. And I will tell you in terms of when UPF1 cleavage occurs, it is before the complete activation of caspases, both the initiator and the executioner caspases, so that we do know that UPF1, and I'll also show you more direct data, promotes the apoptotic process. It's not a consequence of it. We know UPF1 cleavage is mediated by caspases because the cleavage is inhibited by caspase inhibitors, and we know the caspases are casp3 and 7. Max has mapped the major caspase cleavage site to aspartic acid at position 37 in UPF1. And what he's done is expressed the major cleavage product that he sees when cells are treated with doxorubicin. So this is the concentration of the cleavage product. Remember, it's substoichiometric. And measured the degree to which NMD is inhibited. And here, beta-globin TER contains a premature termination codon that triggers NMD. So he's looking at the ratio of the NMD target relative to its normal counterpart. And you can see it increases with increasing amounts of the cleavage product. And he can actually inhibit NMD more and more and more if he adds more levels of this truncated caspase, normally caspase-mediated cleavage product to cells. So this is an inhibition of NMD that is not 100%. How did Max separate the doxorubicin response from the NMD inhibition response? We don't want all the P53 and other types of transcriptional inhibition mechanisms that accompany apoptosis to take place. We just want to cleave UPF1. And so what Max did is he engineered an expression vector for human UPF1 that was active still, but he put Jim Wells' tobacco etch virus proteolytic site in UPF1, and then was able to cleave orthogonally in the absence of doxorubicin, in the absence of damaging DNA, to generate the cleavage product at the levels that he sees with doxorubicin treatment, and then sequence the transcripts in those cells. And this is a really short list of the genes that he found encoded transcripts that were upregulated when UPF1 was cleaved. And you can see that they inhibit the cell cycle, and they are pro-apoptotic in other ways. Max also demonstrated that if he inhibits UPF1 cleavage, he decreases and slows doxorubicin-mediated toxicity. And he found that if he inhibited NMD, so Fabrice Lejeune is a former postdoc in the lab who has his lab in France now, and he has developed an inhibitor of NMD. And so if Max adds the inhibitor to NMD, then washes it out, and then treats cells with doxorubicin, he augments the efficiency and the degree to which apoptosis occurs. All right, what we're probably best known for over the years, because we've worked on it for about 30 years, is the mechanism of NMD, and I'd just like to go over that a little bit. So you know RNA in cells is never devoid of proteins, and the proteins are dynamic on an RNA, often in a regulated way, and that's what I want to show you in terms of the NMD process. We've talked about NMD occurring during a pioneer round of translation, and we define that round as the translation of mRNA that has the CPP80 and 20 cap binding proteins that are acquired co-transcriptionally by pre-mRNA. Here's the exon junction complex deposited in a sequence-independent fashion, but in a 20 to 24 nucleotide position upstream of exon-exon junctions, and here I'm showing some of the other UPF proteins that are stable constituents of EJCs that we have shown. The ribosome is shown here, and when it initiates and then terminates at a premature termination codon in a way that is sufficiently upstream of an exon junction complex so that the EJC isn't removed by the ribosome, then not only do the two translation initiation factors, the two translation termination factors, the eukaryotic release factors bind, but also the key NMD factor UPF1 and its PIC-related protein kinase SMUG1. We did not discover the SURF complex. Shige Ono in Yokohama, Japan did, but we have certainly studied it. Then we know that UPF1 and SMUG1 move to the EJC, at which point UPF1 becomes phosphorylated by SMUG1. There's a remarkable step of translational repression, and then the mRNA is degraded. And I'd like to talk about that step of translational repression, which was discovered by a postdoc in my lab, Olaf Iskin, who now has his own lab in Germany. All right, so here's UPF1 phosphorylated by SMUG1 on the EJC. The repression involves phospho-UPF1 interacting with the two highest molecular weight subunits of a multi-subunit complex called eukaryotic initiation factor 3. And this is a constituent of the 43S pre-initiation complex poised at the initiation codon of this NMD target. What that interaction does is it prevents the 60S ribosomal subunit from joining the 43S to form a functional ADS ribosome. And we know that if we initiate translation from, for example, the cricket paralysis virus internal ribosome entry site, that does not require and involve EIF3 in the initiation process, so there would be no step of translational repression. Even though that mRNA would contain a premature termination codon, there would be no NMD. You need to repress translation for NMD, probably like you need to repress mRNA translation in microRNA-mediated decay before you can get decay. And then we also know phospho-UPF1 recruits the degradative activities that are triggering the decay process itself, or they are the degradative activities, and these include decapping activities, 5' to 3' exon nuclease activities, deadenylating activities, exosome-mediated 3' to 5' decay activities of the body of the transcript, and then I'm not going to talk about an endonuclease, another NMD factor called SMUG6 that targets cleavage endonucleotically a little bit downstream of the premature termination codon. We were really surprised when we found out that this mRNP was targeted for NMD and in fact could be translated, because all of the textbooks said that this mRNP, which has a different cap binding protein, eukaryotic initiation factor 4E, produced the proteins in cells, and that is correct. The most of our cellular protein synthesis comes from this mRNP, the mRNA and associated proteins, but there's a precursor-product relationship, as Fabrice showed, and it is this mRNP that's largely targeted for NMD, and you can see why. It's the one that has the exon junction complexes. By the time EIF4E replaces CBP80 and 20 at the cap, the mRNA has lost the EJCs, and I won't go into that mechanism. But it's even more interesting than that. CBP80 actually promotes NMD. So this is work done by Jungwoo Kwang when he was a postdoc in the lab, and he also has his own lab now in Korea. There's a transient or weak interaction of UPF1 with CBP80, and that allows CBP80 to escort UPF1 and SMUG1 to form the SURF complex, and then escort UPF1 and its kinase SMUG1 to the EJC. Without CBP80, you do not get those interactions effectively. So I think you can see the little dance that proteins do in a very regulated way during the process of determining if an mRNA has a premature termination codon of the type that should trigger NMD, and if so, then again, choreographed steps, the trigger is phosphorylation of UPF1, and the mRNA undergoes decay. Last thing I want to talk about is where in the cell does NMD occur. And I can say that I got a lot of pushback, I think in a kind and compassionate way, more or less, when I said that intron position in a pre-mRNA determines which termination codons in the spliced product that has no introns influence mRNA metabolism. And we had proposed the mark. There must be a mark deposited as a consequence of splicing that is maintained on mRNA until that mRNA undergoes translation, because we knew NMD was a translation-dependent mechanism. And the other thing that people had a little hard time wrapping their heads about was that NMD, yes, it targets largely newly synthesized RNA, but we were saying it was nucleus-associated. I never said it was nucleoplasmic, but nucleus-associated. We don't believe there's translation in the nucleus, and if there is, it's a small amount, and it's very hard to prove. Certainly there's translation in the cytoplasm. And so what we meant was that mRNA that co-purifies with nuclei, the nucleus in which they were synthesized, could be translated and therefore degraded, provided they had access to translation by cytoplasmic ribosomes. And so I would go to a meeting in Stockholm all the time, and one of my favorite guys was Bertil Danaholt at Karolinska, who I know he's now retired, but he was able to study, after we had our proposal, that the large Balbiani ring granule of Chironomus, it's an insect, Chironomus tentans, that he could visualize using electron microscope tomography, was invariably exported from the nucleus to the cytoplasm, 5' end first, here's the 5' end, and at a point where the 5' end became associated with ribosomes, before the 3' end had yet to cross the nuclear pore completely. So this little blob is the nuclear pore, and maybe you can see the two membranes of the nuclear envelope. And so I loved this, because this is how I thought about where nucleus-associated NMD was occurring in cells, and I would talk to him every year, whether I saw him or not. I would contact him and I'd say, do you think that mRNAs on the way out could also contact the cytoplasmic fibrils, filaments that extend from the pore? And he finally had data in support of that. So there are many chances for an mRNA, newly synthesized mRNA, being exported from the nucleus to the cytoplasm to be translated in association with the nuclear envelope. And then another person who I kind of bugged forever is a good friend of mine, Rob Singer, who has the ability to look at single molecules. And finally, the biophysics and the mathematics and his ability to do microscopy developed to the point where I was able to send a postdoc in my lab, Hana Sato, to work with one of Rob's graduate students, Tatiana Trek, and the two of them were able to actually visualize where nucleus-associated NMD occurs in cells. And so I just want to go over one experiment. What they did, among many, is to generate a bidirectional promoter that was Pane-inducible, so can be rapidly turned on. And one promoter produced globin mRNA that was PTC-free, and the other produced globin mRNA that had a termination codon, a premature termination codon that triggers NMD. And they both gutted the 3' UTRs of globin mRNA and replaced them with beta-galactosidase sequences from E. coli that were different. And then they could anneal antisense 60 nucleotide oligos that were fluorescently labeled. So the LAC-ZA could have been red labeled and the B green labeled, or vice versa. And then they were able to track, and these are in osteocarcinoma cells, the individual mRNAs while they were in the nucleus, being exported across the nuclear pore and in the cytoplasm. So this is a ratio of the NMD targets versus their normal counterparts. And you can see DAPI staining defines the nucleus. The nuclear envelope is defined by the red green antibody that recognizes the nuclear pore complexes that are all across the nuclear envelope. The ratio in the nucleus is bouncy, these are hard experiments, but more or less constant. That ratio is higher than the ratio in the cytoplasm, because remember, when an mRNA gets released into the cytoplasm, it has a half-life just like its normal counterpart, according to all the data that we had from the mid-90s. And there is where NMD is occurring when you track single molecules. It's on the cytoplasmic side of the nuclear envelope. So I am very happy to say that we were also right with that. And this just comments on there being no change in the ratio in either cellular compartment, and that NMD occurs presumably while the RNA is still associated in some capacity, maybe with the cytoplasmic filaments on the way out to the cytoplasm. So what we can conclude from this work is that NMD is a rapid process, and an NMD target undergoes biphasic decay. Once that mRNA hits the cytoplasm, NMD occurs within the first minute, and our best estimate is anywhere from 2.9 to 56 seconds. I know that sounds like a broad range, but it really isn't at this point in our technology. And then those mRNAs that escape NMD, because NMD is never 100% efficient, degrade with the other phase of its decay profile, and that is with a half-life of 12 hours that is equal to the normal counterpart. So what have I told you? I've told you that NMD is an important quality control pathway in human cells. It eliminates mistakes routinely made during the expression of our genes. And it also protects carriers of a single PTC-containing disease-associated allele from disease, where the other disease is normal. So it prevents the production of truncated proteins that you need from only one allele in order to have a disease. NMD is also used by cells to autoregulate many RNA binding proteins. I gave you the example of polypyrimidine tract binding protein, and it does so in a process called alternative splicing coupled to NMD. There's also alternative 3' end formation coupled to NMD is another variable. And additionally, NMD targets 10% of our normal mRNAs. It's a way to regulate, cells regulate the pathway, usually inhibiting the pathway in all the examples that we have so far as an adaptive process. And we have done, there were 3 instances where the mechanism is known. We've done 2. Apoptosis you've heard about, where UPF1 is caspase cleaved to downregulate NMD when the efficiency of NMD is inhibited to promote apoptosis. We've also shown that during myogenesis, the efficiency of SMD is upregulated, and this is because the efficiency, did I just say NMD? The efficiency of NMD is inhibited because the efficiency of a competing pathway we have discovered that I haven't talked about is in the opposite direction. And then interestingly, Miles Wilkinson has shown that during the differentiation of neuronal stem cells to neurons, the efficiency of NMD is inhibited, and it's inhibited not by caspase cleavage of UPF1, but by upregulation of an mRNA, a microRNA that degrades UPF1 mRNA. And so I've also told you that we have defined a nuclear process that can mark newly synthesized mRNAs in a way to affect cytoplasmic metabolism. I've told you CPP80 and CPP20 bound mRNAs can be translated for the purpose of quality control. We've talked about there is nucleus-associated mRNA translation, and then our studies have gone on to what almost half of the lab studies, and that's a Stauffen-mediated mRNA decay pathway that at one point converges with the NMD pathway. And in this pathway, we've learned new roles for long non-coding mRNAs. That should be long non-coding RNAs and mRNAs by they're containing ALU elements, which are repetitive elements. We have 1.6 million copies in our genome. And with that, I've pretty much acknowledged everyone as I went along. Here's some pictures of current lab members, some of whom are here today, and thank you for your attention.